Thank you very much, and sorry I cannot be there in person. It's very real pleasure for me to present. It's, I guess, the first time I present a conference in Florence. That is my alma mater, so it's really exciting. I want to tell you some of the things we've been doing recently on using machine learning for learning biophysical model. Biophysical also, we started now with introducing some machine learning for quantum systems. So this is my group, before I forget, and I have available positions for postdocs and graduate students. So I'm hiring. If you are interested, or if you know interesting people, let me know. Let me start by stating what we want to do, what our final goal. You know, last summer, there was a big splash in the news because AlphaFold 2 from DeepMind managed to predict quite accurately a lot of folded structures from sequences. And if you think about it, what it does, it gives you the most stable point on a free energy landscape. If you believe in the thermodynamic hypothesis of protein folding, AlphaFold gives you the minimum free energy in a free energy landscape. That is a very important point, but it's not the full landscape. It's just a point. What we would like to do instead is to try to have an idea of the whole energy landscape. Because if we can get the full energy landscape, we can understand function. Here is a recent result, not from my group, but from a collaborator, that shows a protein, the dynamics of a protein performing its function. Here you have on the left, you see the different states in a Markov state models, and the protein moving on the right, and while it moves, it jumps into different minima of this landscape. You can think about these different circles at different minima in very complex energy landscape. So it's important to understand not just one point, but a little bit more about this energy landscape. And as I said, if we can understand the overall thing, if we can have a way to map the overall thing, we can understand function. That means we can develop vaccines, enzymes, drugs, understand disease, and so on. Of course, this is easy said, but not easy done, because of course there is, if it was easy, everybody would have done it. The main challenge is the fact that proteins in general, large biomolecules, have a very large range of time scales. They go from picosecond time scale, where you have local motion, local and fast motion, to up to, if you're interested in folding, you start to have folding events for fast proteins at microsecond, but they can go up to minutes and beyond. And if you want aggregation, is even slower. So this is impossible with brute force simulation to put in all the details and and just watch what's going on. You cannot map, you cannot sample the overall energy landscape with molecular dynamic simulation at all atom details if you want to get relevant biophysical processes. However, we know from statistical mechanics that when systems are large, you don't need all the details. Sometimes doing some smart averages can take you a long way. And I don't want to discuss here all the evidence for this, but we believe that we can develop minimalist models that can still capture the relevant physics and give predictions, even if they don't keep all the atomistic details. Of course, this cannot be just an ensemble average, because these systems are very heterogeneous. So we need to have some smart way of averaging. And we want to do it at different resolutions. So this is coarse-graining, and usually it consists of two steps. The first one is to decide the mapping. That is to decide what we want to keep. And this is already an important problem, and we are working on it, and we have a preprint coming up soon, but I'm not going to discuss it today. What I want to discuss today is the second step. That is, once you have the resolution, once you have decided the resolution, which details to keep, you want to associate an Hamiltonian, an effective Hamiltonian. It's not an Hamiltonian given from first principles, it's an effective energy function that can reproduce some properties of your system. And usually, historically, this energy function have taken the functional form borrowed from all atom energy function, from force fields, like with local terms and non-local terms. This is from an old paper we published more than 20 years ago. But there is no reason a priori why this effective energy function should have this functional form, because it's not a first principle Hamiltonian. It is a Hamiltonian that is effective. It needs to reproduce some final results, but it's like a normalization of the first principle Hamiltonian. And the way to design such an energy function depends on what you want to preserve. If you want to reproduce experimental observation, that is macroscopic averages, then it's a top-down approach and you'll follow certain procedures. If you want to reproduce the thermodynamic of atomistic models, that is bottom-up, and you follow a different approach, or there are other ways, like you want to reproduce some structural properties. And in this presentation, I'll tell you both about the bottom-up and top-down. I'll start with the top-down, and then I'll tell you about the bottom-up. Of course, eventually we would like to merge the two together. And then I'll tell you how what we have done in coarse-graining over the years, we're now porting it into understanding quantum nuclear effects. So let's start with the first part. A few years ago, we developed an approach that is called ODEM, Observable Driven Design of Effective Molecular Models, that takes global observables from experimental data, like free energy or fret measurements or NMR data, and you'll see later some examples, and try to define a coarse-grained model to reproduce this data. Here we use the fixed functional form, but this can be a neural network. There is no reason for this to be you just need an Hamiltonian, an energy function with a lot of parameters. These epsilon are the parameters. And then you want to tune the parameters, again, can be the weights of a neural network, such that you can reproduce this, the global observables. So you start with the initial guess of the parameters and the experimental observables. You run an initial simulation with the initial set of parameters, and you compute the observable, you compute the equilibrium distribution and the observable from simulations. And then you define a likelihood that tells you how the measure observables in simulation match the observables obtained from experimental data, from experimental measurements within the error bar. And if you have many observables, this become the product of many exponentials. So it's if just one of them deviates farther, this likelihood goes down quite quickly. But this likelihood is characterized by the fact that it's always between 0 and 1, as it should be. And beside having the maximization of this likelihood, beside giving you the model with the best parameters, with the parameters that best reproduce the experimental data, also quantifies this agreement with the number between 0 and 1, so you can decide if you can trust this model or not. So you can at this point, you can maximize this likelihood, select a new set of parameters and iterate. And you can do this because these coarse-grained models are quite cheap to simulate, and so we can repeat these simulations many times. This was the first application of this method that we published in the paper, and this in order to disentangle the problem of the error from experimental data and the possible problem in the model, we generate experimental data synthetically. So we call it synthetic threat. We generate experimental data from all atom simulation. And then we train the Coarse model to reproduce it, to reproduce these global observables. And as you can see here, the initial model was not very good. These are distance distributions, so like they would be, they are the proxy for threat distances distribution. The initial model was not very good. These two peaks here are for the folded and for the state, and the initial model couldn't reproduce it. But if we optimize it, we can get very close to the reference. That is what we get from the all atom simulation. And then we can cross-validate it on observables that were not used in the training. For instance, these are, again, proxies for tryptophan fluorescence. And you see that the model is significantly improved and much closer to the reference with respect to the initial model, in particular for the transition state. So now this was a proof of principle. We were happy about it. And now we are working on real experimental data. So here you can see the NMR data. What we did with the NMR data for a carbon alpha, carbon beta model ubiquitin. We use the carbon alpha, carbon beta. So not just we take the carbon alpha model. We can take the carbon alpha atom of each residue, and we also take the carbon beta model if it's not glycine. And we use the scalar spin-spin coupling constraint. And you can see here, after many iterations, the likelihood converges, and you get an agreement with the experimental data that is comparable to what you get with atomistic force fields, if not better. And more interestingly, now we are using free energy differences as experimental data. We use an approach that we proposed almost 20 years ago, but with this new ODEM maximum likelihood method. So the idea is to reproduce mutation as a perturbation. Experimentally, you can mutate a residue, usually is conservative mutation from a large to a small residue, and you consider the difference in energy upon this mutation as a perturbation, then use perturbation theory, and you can use the delta free energy. Usually as delta G is the difference in free energy of the stability. The difference in stability or the difference in free energy at the transition state. So we use these for two proteins, ubiquitin and protein B, and we use only a carbon alpha model, and we use the native and non-native interaction. Okay, these are just details so on the representation. And for ubiquitin, everything works very well. You see here, you have the final results, the experimental data, delta G is the free energy differences for mutation, experimentally measurement, and the calculated ones. The initial model is in blue, and using a model with only two body interactions, you get already a very good agreement. This is a very simple model with only two body interactions, like Lennard-Jones kind of interactions. But when we go to protein G, the situation is different. The initial model is in blue. And then when we used two-body interaction only, there is not, there are three residues, these three residues here, that still are far from the experimental data. The calculated values are far from the experimental data. And these three residues here, these are the three mutations, are highlighted here in the protein. And they are exposed to the solvent. And this makes sense because we are using two bodies on interaction, and these residues are exposed to the solvent. It's clear that the solvent needs to be taken into account somehow, and two body interactions cannot do it. So we added multi-body terms in this form, but this is not, again, can be a neural network. And we get a significant improvement on the agreement. And so this is a clear sign that multi-body terms are essential to reproduce the agreement with experimental data. This is a top-down proof that you need multi-body terms in that is simple force field like in an atom simulation doesn't work for a Coarse-graining model, and we'll see this later also for a bottom-up approach. So this was for the top-down. Now we are also at the same time we are pursuing a bottom-up, and as I said, eventually we want to start with the bottom-up and continue with the top-down to correct with experimental data. And in the bottom-up, we use the thermodynamic consistency. Thermodynamic consistency means that we want the free energy of the Coarse-graining model to match the free energy of the atomistic model when projected on the same coordinates. And if you get the free energy, consistent the free energy, you get consistency in the full thermodynamics because we are in the canonical ensemble. So what it means, this is an example of consistency, very simple toy model going from 2 dimension to 1 dimension. You coarse-grain away the y dimension, and you can do this analytically, because what you need for thermodynamic consistency is this set of equation. What it means is that the effective energy function for the coarse-grain model is fixed by this expression. So is given by this expression, is given by the logarithm of the marginal distribution of the atom model. So in principle, the problem is solved because you have an expression for the effective energy function that gives you thermodynamic consistency. In practice, these integrals are impossible to solve because these are extremely dimensional. You go from millions of degrees of freedom to maybe hundreds of degrees of freedom, so you cannot do this integral, and so it's impossible to calculate. Here we could do it because it's in 2 dimensions, but in practice we cannot do it. Fortunately, many people like Greg Voth or Will Noit have reformulated this thermodynamic consistency variationally, and they showed that it's possible to define a functional. This is the functional here, and the energy function U here that minimizes this functional is the same as the one that reproduces that satisfies thermodynamic consistency. So this can be used variationally, and it was used in the past. If you put a functional form for U with a lot of parameters and you minimize it. But again, a lot of the functional forms that were used were 2 bodies, and that don't work. I just showed you that we need multi-body terms. And so instead of restricting a functional form to 2 bodies or try to correct with 3 bodies, we decided to put a neural network for the energy of U, so that you can span a much larger, in principle, you can approximate, can go close enough to any function if the network is expressive enough. And then we have used until recently as a test system this small protein signaling that is 10 residue long. It's very small, so it allows for fast testing, but also is already with a rich landscape, because even if it's only 10 residues, when you simulate it in an atom in water, it has 3 minima: a folded state, an unfolded state, and misfolded state. So this is a very good test case. It's fast and is a rich landscape. So this is what we can do with this approach. As you can see, we can learn an effective functional form for this protein. This is the atom reference, and this is what you obtain with the Coarse-graining model with carbon alpha only. And they match, I would say, pretty well. There is maybe the unfolded state is a bit overstabilized, but there is some variation in the results. If you do it many times and you average, if you do like a consensus model, it performs better. Anyway, so this is the state of the art of what we can do with training for one particular system. But what we would be interested in is to train a transferable model. This is because it doesn't make too much sense to train a coarse-grained model on a system on which you need already all atom data to define the model, because then you already have a sample with all atom details and you don't need the coarse-grained model. Because what I didn't say is that here, this F that appears here is the force of the atom model projected into the coarse-grained coordinate. So in order to use this approach, you need to have saved all the forces of your frames in the atom simulation. So you have to have a pretty good sample of all atom configuration with forces. And so this was a proof of principle showing that it works quite well. But what we really want to do is we want to develop a transferable coarse-grained model. That is, we want to take small systems like peptides, and this we're working with a database of many octapeptides, peptides of 8 residues, with different sequences in explicit solvent. We can run, when these systems are small, we can run a lot of simulations. And then we learn a universal force field, meaning we learn a force field that depends on the amino acid species, on the bead species of our model. And then we can compose it into something larger that we cannot access with the atom model. And this is the neural network architecture. Is based on the SNET that is developed by Tachenko and Muller a few years ago. Anyway, if you are interested, I can go into the transferable architecture later. But this is where we are now. We can do it. This is again on our Chignoline test system. Chignoline is 10 residues, so is larger than the octapeptide that were used. Octapeptides are 8 residues. So not only this sequence has never been seen, but this system is really, the network is really extrapolating to a larger system, larger than the data was trained on, and completely different sequence. And indeed, it can reproduce the 3 minima of Chignoline, folded, unfolded, and misfolded, with structure that match very well the atom structure. And we're still working on this, but we are very excited about these results. Another thing I want to quickly discuss again is these multibody terms that we believe are essential to make these models work. So we decided to have a look at these multibody terms to understand if this is if a neural network is really required, or we could get away with some simpler function. So we defined a neural network that is in practice is a multibody, many body expansion. We started with the 2-body only network, and then we added the 3-body correction, and then a 4-body correction, and so on. So basically many body expansion. And this is the results. If you look at the result at the table on the right, you see the Kullbaum-like Liebrecht divergence for different models. The CGNet is what we obtain with the full neural network. And with 2-body, it doesn't perform well, clearly. The error is much larger than CGNet. 2-3 bodies is better, but 3 bodies are not enough. And basically, the story is that you need to get up to 5 bodies to get to comparable performance as using a full neural network. So this was kind of a surprise for us, the slow convergence of this series. But and again, it underscored the importance of multibody terms and complex multibody terms. So for the moment, we stick to use neural network until we can understand more about the nature of these many body interactions, how we can model them with functional forms. We'll stick to neural network because it's basically impossible to come up with decent functional form for all these many-body terms. And this is the result of what you get in the multibody, in the many-body expansion. This is the reference CGNet. With 2-body only, you don't get, you just get a blob. With 3-body, you start to see the formation of the 3 minima, and you get more and more quantitative. This is the reference here. This is the reference all atom, and you get very close to the all atom model if you use up to 5 bodies, 2, 3, 4, with C is for chirality, and 5-body terms. So this confirms the results from the top down that we need multi-body terms. Now, quickly, in the few minutes I've left, I want to tell you about we can use these same approaches in a quantum setting, in particular to take into account nuclear quantum effects. So nuclear quantum effects, even if we have, you may know that a lot of more modern force fields for all atom force fields are now neural network force fields that are trained in order to capture the electronic properties. You normalize away, it's still a kind of coarse-graining, you normalize away the electrons, and you have a force field that depends only on the position of the nuclei, but takes into account the effect of the electrons. But even if you have this, you still have to, if you want to be accurate, you still need to solve the Schrödinger equation for the nuclei. And for many systems, this is not so important, but when you have hydrogen, it's important because hydrogen is light. And for instance, here in this figure, you see the localization of hydrogen and the difference between hydrogen and deuterium when you take into account properly the nuclear quantum effect, and is different from the classical case. And there are a lot of cases where is important. And in biology too, for instance, protonation, deprotonation, and so on. So in order to do this, we decided to use the Feynman path integral, the quantum classical isomorphism. And that means you may be familiar with the Feynman path integral. Basically, in practice, it means that each degree of freedom, each atom gets associated to a ring polymer. All these beads in the ring polymer are replicas of the atom, and they are coupled together, and they interact with each other with the copies of the same atom. They are interacting with each other through springs. They are coupled with springs, and copies from different atoms interact with interaction potential. So you can reformulate the quantum problem into a very large dimensional quantum problem, a classical problem. And this is exact in the limit of the number of replica going to infinity. So we make the problem much heavier. It's classical, but it's much heavier computationally. However, we thought this is the polymer. We can coarse-grain it back to a single bead. So we perform some initial path integral simulation with these ring polymers with many beads, and then we coarse-grain it back to one bead that is going to be a potential that is different from the classical because it incorporates effectively also the quantum nuclear effect. And we call this approach PIGS, a centroid path integral coarse-graining simulation, because it's this idea that of the we like this analogy with the idea of the ring normalized back to one point. And it works very well. Here I'm going to show you a few results. So this paper is now on the archive, is under review. This is for the benchmark is the OH bond that can be modeled with the Morse potential, and the Morse potential can be solved. The Schrödinger equation for the Morse potential can be solved analytically, so it's a good test case. And here you see the reference, the quantum mechanical results at different temperature, and here you see our results. at different temperature. And you can see that the peaks correspond exactly. This is these are the spectra of the OH bond. If you use an alternative method like centroid molecular dynamics, it doesn't. You have a shift in the peaks. It doesn't work because of several problems with centroid molecular dynamics that I can discuss later if you're interested. So in order to make it work, we have this approximation, this temperature elevation approximation. Again, this is maybe a technical detail, but I can discuss it more later, and takes into account of this shift in the centroid molecular dynamics. Basically, what we say is that the landscape that we're learning an effective landscape for this one bead that includes the nuclear quantum effect. And this is independent of, the energy landscape is temperature independent. So we learn it at high temperature and then we use it at low temperature. And it works. And this is for the full water molecule. And you see again, in red is our results, and in black, in gray, is the most accurate quantum simulation we can do as a comparison, and they agree perfectly well. But in addition, we can go to very low temperature because of this trick that we have learned, the energy surface. And so we can go to cryogenic temperature with good agreement with experimental data. And this cannot be done with any other method because this number of replica in the ring polymer goes to infinity as the temperature goes to zero. So it becomes impossible also with this path integral formulation. But in our model, we coarse-grain back to one bead, so we don't care. And this is for bulk water. Again, in red, our one bead effective potential simulation, and in black is the best quantum mechanical solution we could compute quantum mechanically. And again, you can go to cryogenic temperature, and it agrees with the limit that should go to at zero temperature. So with this, I want to summarize what I discussed. Basically, coarse-grain model, using a coarse-grain model to capture the physics of complex biomolecules in a simplified manner, but taking into account important effects like quantum, multi-body terms. We can do it with machine learning. And what we've started to do recently is to also use it and to take into account nuclear quantum effects. And the ultimate goal is to combine the top-down and bottom-up. So the idea is to start with the bottom-up with atomistic simulation and then correct with, because eventually the comparison is with real experimental data. I want to correct with experimental data. And with this, I want to again thank the people who have done the work, and all the funding sources, and all of you for your attention. Thank you.
Thank you very much, Cecilia, for this beautiful talk. So do you have questions or any requests for Cecilia?
Hello, Andrea, nice to meet you. I guess my question applies mostly to the first part of your talk. So the coarse-graining potential is a sort of effective field theory, no?
Yeah.
We call effective field theory in particle physics. And to make predictions out of that, you must construct it with a theoretical logic that allows you to simplify, to truncate at some order, right? In normal approach. So to what extent, even if you fit your potential, like you're doing in the first case, to the data, then you would be entitled to expect that even for the same system, you will be able to make other predictions of different observables. And even less, how can you port this to another system? But this, I guess, is what you are instead addressing in a second part of your talk.
So this is a very good question, and indeed you are right. We need to, what I didn't discuss is in this architecture. Let me go back. Here, there is this prior energy here. That's very important. Without this, I don't have the figure here, but without this, nothing works. This is all the physics constraints. These are the asymptotics. We need this energy. So this is a prior. And it ensures that the asymptotics are correct. That means two atoms cannot overlap. A bond cannot be stretched infinitely or compressed infinitely. There are angles cannot be again stretched or compressed too much. There are dihedrals, and with the chain. Now we are also using some other terms, but these all the physics, the symmetries, all the physics, you know, you need to put it in this prior. The more, the better. And if you don't use the prior, nothing works. You cannot, not only you cannot extrapolate, you cannot even fit one molecule.
Okay, thank you.
Questions?
Thank you. Hi, Cecilia. Eliška here. Thank you very much for the interesting talk. I actually had like a bigger picture question about this usage of a quantum effects. I have a feeling that in these biological structures, the underlying quantum effects sometimes lead to like a controversial discussion. So I wanted to ask you, what's your opinion on like a validity and scale of these effects, and for which specific processes do you think it might be useful, and so on? Thank you.
Yeah, so we want to use it first. What we are doing now, we want to use it on water cluster. That is a very hot topic right now. A lot of people are studying confined water, water cluster, and water is a system that depends where the nuclear quantum effects are very important. If you, I don't have the classical case here, but the classical prediction will be completely off. You cannot predict the spectrum of water without nuclear quantum effect, because there are two hydrogens that are very light. And so it's not clear how this will affect confined water, water clusters, and so on, that are important, not just biologically, but also for industrial application. This is what we're doing now. But eventually, we want to use it in biological systems. For instance, we are interested in protonation. You cannot describe protonation with the classical force field. It's a quantum effect. I mean, it's a quantum effect. So you need to have the possibility of delocalizing a proton. So there are several processes where you have protonation that is important in biological systems. So this is one example.
Okay, thanks a lot. So I'm afraid we have to move on, because otherwise we're going to be late for lunch and Giuliano won't be happy. So I thank Cecilia again for her time and for this beautiful talk. So,